Hey, what's up guys? This is Adi from Shastri Nation, and today we're going to be talking about biomolecules again. Well, this is part 2 of the video. If you haven't already seen it, go ahead and I'll leave the link in the description, or you can click right here and you can watch the video. Because we will be building off of the concepts learned in the last video. So anyways, we've talked about biomolecules. So biomolecules are the things that make up us, like proteins. Proteins. Sorry, I almost said amino acids. But proteins, carbohydrates. Lipids. And DNA and RNA, or nucleic acids. Nucleic acid. So basically in this video what we will be doing is we will talk more in depth about each of these biomolecules. So let's go ahead and start with protein. Proteins. Let me go ahead and change the pen type. Yeah, that's better. Okay, so let's go ahead and talk about proteins. Okay, so proteins are, again, made out of, the monomer is amino acids. And so I'm just going to use an AA as the amino acids. It's just shorter, and so I'm just going to use that. I don't know if your biology teacher does that, but I'm going to use it. And the polymer is polypeptide. Polypeptide. Okay, so what are proteins? So proteins are basically what govern us as living things. Proteins are used in enzymes. Proteins are actually enzymes. Muscles, they are used in muscles. So how these work is that they contract and expand to let us to move, you know? And sorry, I didn't explain enzymes. Enzymes are basically the things that speed up every single reaction in our body, and that's basically converting everything. That basically controls everything from movement to digestion to thinking and everything like that. Well, not exactly thinking, but everything really it controls. And so since enzymes control all the reactions, and they're made of proteins, proteins technically control us. And let's see. So basically, enzymes. Sorry, let me get a new color. Proteins are made up of amino acids, and amino acids, there are 20 different types. There are 20 different types of amino acids. And this is a reason why scientists actually thought that proteins were the genetic code. Thought by scientists. Oh, sorry. Yeah. Okay. So they're thought by scientists to be the genetic code that governs us. I mean, it makes sense if you look at it. There's 20 different types of combinations. I mean, there's 20 different things, and so there are way more combinations compared to only 4 in the DNA. But this is obviously wrong in nucleotides. DNA is actually the thing that the genetic code that basically controls everything in our life more than proteins, and you'll see why. So the protein is really made up of amino acids, and so the basic structure of amino acid is like we've talked about in the last video, say you have a nitrogen, you have some carbons, an R1 group, and you have a the carboxyl group here, and then you have a hydrogen again, and then an amine group right here. Alright, so let's go ahead and label the parts. True, I mean, I'm not going to just leave you to that. So the green would be the amine group. And, you know, you'll just need to memorize these really. There's no real way to tell. It'll just make sense, you know. So I know that since there's a nitrogen in this and two hydrogens, that it's a amine group. And then, so the red, sorry, let me go ahead and write that. So amine group. And so that is called the N-terminus side. Okay, and then so we have the, in blue we have the carboxyl group. And that is called the C-terminus side. Okay, so what happens is like you have the C-terminus side always bonds to the N-terminus side. So these sides always bond. So like if we had another molecule, the N-terminus side or the amine group would bond with this. And that's just how this works. Sorry, this is O and then H. Oh, let me go ahead and erase that. Oh, I wrote this down wrong. Okay, so it's a carboxyl group. So this is an O, and then you have another H here. And that would be the carboxyl group, sorry. And then so, as you know, since the formation of polypeptides or any polymer, it's the process called dehydration synthesis, and you have the 2H2O here. Two H's and one O, and that leads to water being a product. Okay, so let's go ahead and talk about the structures. Let me get a good color here. Okay, so let's go ahead and talk about the levels of structures in a protein. So you have the primary sequence. So the primary sequence is just the order of the amino acids, order of AA. The secondary group, secondary sequence, structure. This should be structure too. Okay, so the primary structure is the order of the amino acids. So the secondary structure would be the interaction between the primary structure. The interaction between the primary structure. And then we go to the tertiary structure. I'm just going to abbreviate structure. Yeah, you're going to see I use a lot of abbreviations in my science classes and stuff like that. And, I mean, it's fine if your teacher doesn't mind it. So the tertiary structure is the interaction between those. So as you can probably tell, it's just like that. And then we have the actual protein. So the protein would be the interaction between those. So this is the levels of a protein. Now proteins are 3D structures, so they're 3D structures, and you can tell that maybe even a single amino acid change could completely change the way a protein reacts with other things and could completely ruin something. And you'll see how that could happen in my whenever we get to mutations and stuff like that. Okay, so let's go ahead and talk about... Sorry, I'm just gonna... Okay, let's go ahead and talk about lipids. Lipids. Alright, so lipids are pretty self-explanatory. There are fats and oils. So again, the monomer is kind of weird. Again, but it's a fatty acid for some of them. See, the lipids are weird because they can fit into other groups, but they're at the same time they can fit into other groups. They are also different. It's kind of weird, I guess. And so what lipids are is just they are a if you remember from the last video, we talked about the hydrocarbon, which is the most basic organic structure. So it looks like that. And what lipids are basically a longer version of this. Longer version. These might be very, very long, like 10, 20 carbons long, you know? Or even more than that. And so again, we have a carboxyl end. If you remember from the last, when we talked about proteins, it's right here. The carboxyl end would be this end right here. Let's just go ahead. Yeah, so this would be the carboxyl end. And the carboxyl end, alright, here we are. Carboxyl end is hydrophilic. The carboxyl end is basically hydrophilic, which means that it likes water or can be dissolved in water. Or just remember it as it likes water. And so you can tell that by looking at the suffix here, meaning saying philic, which means liking or loving. So if we had a hydrophobic molecule, that would be water hating. So we look at water and then we look at phobic, meaning it's hating. Water hating, water liking. Okay. So the reason, for example, if we put oil or something like that inside of water, you know how oil doesn't dissolve into water? And that is mainly because of the lipids long structure. Because otherwise it would dissolve into water because it's hydrophilic on the ends. At least that end would dissolve, but since it's so long, it doesn't dissolve. And that's why you can never dissolve oil in water. So again, the polymer, if you don't remember from the last video, again, if you want to watch that video, it's down in the description below. Anyways, the polymer of a lipid is the triglyceride. And so this is just like a chain of lipids, basically. Oh, sorry, this is a chain of fatty acids, my bad. And so one place you can actually see this in action, or where lipids are actually used, is in the cellular membrane, the cell membrane, in animal cells and plant cells. So the cellular membrane is made up of these things called lipid bilayers, phospholipid bilayers. And so these look like this, but they're, they might be like a whole lot, like this. And we'll learn more about this when I make my video about the membrane, the cell membrane, and stuff like that. But you get the point. This goes all the way around the cell. And you have your nucleus and other things inside of the cell. And so these are all called phospholipids. And so this is called the head of the phospholipid, and this head is hydrophobic. Hydrophobic, and the tails are hydrophilic. And so that is what lipids are used for in the cell. Well, lipids can also be used for long-term storage. So lipids long-term energy storage. Alright, and we go to carbohydrates. So carbohydrates are pretty similar in structure, at least, and overall function to lipids. So carbohydrates also store energy, but not as much as lipids do. Lipids store a lot of energy. Okay, so some examples would be. Sorry, let me go ahead and write that again. I just noticed how badly I wrote that. Alright, oh, my bad. Store energy. Okay, and some examples would be sugars, like glucose. If you remember our my video about cellular respiration, and starches. And so basically, it's just a long carb. If you remember the formula for glucose, it's C6H12O6. And so, you had carbons, hydrogens, and oxygens in them. So basically, there's a little formula that you can use, and most sugars or carbohydrates go by this. And that is C2H. Sorry, my bad, 1 second. It's not C2. Let me go ahead and write that again. Oh, okay. So it would be C. Let me get parentheses. CH2, H2O. And then you have N. So N would be the number of each of these. And so for every one carbon, there's 2 hydrogens, and then for every carbon, there's 1 oxygen. So as you can see, this applies to this because there is, since there's 6 here, there's 6 here, and then there's 12 because it's times 2. So it's a neat little trick to figure out if it's a carbohydrate. So again, if you don't remember from the last video, let's go ahead and make a bar here. The monosaccharide. The monosaccharide is sorry, the monosaccharide is a monomer. Kinda got those switched up, but yeah, I was gonna say that's the monosaccharide is the monomer, but yeah, the monosaccharide is the monomer, and the polysaccharide is the polymer. Yay! Okay, so basically, again, like these add, these make up these, and then through dehydration synthesis. Yay! Okay, and so another cool trick of seeing if we have a sugar in our studies or science is to look at the suffix. Oh yeah, I forgot to mention about enzymes, so we'll talk about that later. Okay, so, but if you see the suffix -ose, like glucose. If you see that, let me go ahead and box that. Ose. That means that it is a sugar. It is a sugar. And so, it's just easy ways to figure out, you know, what things are. Okay. Okay. Okay, let's go ahead and talk about nucleic acid. Nucleic acid. Alright, so our last biomolecule. Yeah, this has been a long video. We're almost done. This isn't too bad. Okay, so this makes DNA and RNA. Okay, so DNA and RNA is basically the structure that govern everything in our lives, from, you know, how we look to, you know, anything really. How, you know, what color our eyes are, or how many, you know, what color our, you know, body is, to whatever you can think about. This is what controls it. Anything about yourself, this is what controls it. And so these are made up of nucleic acids. Well, these are made up of, these are made up of nucleic acids. And then the monomers of this would be a nucleotide. Okay. So the nucleotide, there are 5 really nucleotides. One in RNA, but in DNA it's G, guanine, adenosine. Sorry, adenine. I keep saying adenosine triphosphate from glycolysis. That's not from glycolysis. I don't know where it's from. I don't know. Okay, but adenine, cytosine, and thymine. And then this is all in DNA. Sorry, let me go ahead and get a new color. And then, so this is in DNA. And then in RNA, so RNA, we have the molecules, we have the same molecules, but instead of, okay, so let me go ahead and write their base pairs. So it would be C, and then instead of A goes with T, you'd have A goes with U in an RNA, and then you would have G here, and then you would have A. So this goes with this, and we'll learn more about this in our video about, we'll learn more about this in our video about DNA and all that kind of stuff, you know. Yeah, we're not even close to being done with everything about biology. So, I was saying that our DNA really does govern everything of us, in essence. And so, you know how I said that proteins really govern everything, like, you know, they control every single part of our body? Well, these are the things that control proteins. They control how proteins are made and stuff like that. And so you can see that this is a real mastermind behind all of life. So these instructions, they give the instructions to make so these are very, very, very important. Let me go ahead and write that again. Okay. I'm trying to make this fast so I can let you go. So these are the instructions that make to make proteins, and proteins are made in ribosomes, and you know, we'll talk more about all of this stuff. it's really interesting to see what things actually are in our body, you know, how we get these things and stuff like that. So yeah, let's go ahead and talk about the structure of a nucleotide. Alright, so one nucleotide is like a, let me get a new color. So we have like this thing, this structure like this, you know, and we have it go down like this, and then these two connect. And then so at every single quarter we have a carbon. Well, except for this. This one has oxygen, so see, carbons. Okay, and then so this carbon attaches to the phosphate group, basically. So we have the CH2O, and then this attached to the phosphate group, the phosphate group. So a P in a circle means phosphate group. So this means phosphate. And then this would be attached to the base. So you might be saying, hey, that's very close to, you know, you might be saying, hey, that's very close to, ATP. And I'll answer you, yeah, it is. It is very close to ATP. So this would be the DNA structure, and so this would be the base would be the sugar would be deoxyribose. Deoxyribose nucleic acid instead of ribose nucleic acid. So this would be deoxyribose, would be ribose, and so RNA would actually be protein. And so you'll see that you'll have two phosphates, and then you'll break off a phosphate group, and that's the energy needed to put in a nucleotide and make. This is used during transcription and translation and stuff like that. So before I keep rambling on about DNA and everything like that, I would like you to I want to let you go, and ooh, you saw the gray. Cool. Alright, anyways, I just wanted to let you go, because I could talk about this for a long time. And by saying that, I keep letting you stay here. Wow. Okay. Anyways, thanks for watching my video on Biomolecules Advanced. And I hope it helped, really, because that's why I do these. And if you have any suggestions, let me know. I have a little Google form that you can fill out in the description box, and you can go ahead and fill that out. And if you watched this without watching the first one, none of this probably made any sense. And so I wanted you, I suggest you watch the first one, because all of this would make a lot more sense. And probably for the next video, I don't know what I'm gonna make. I'll probably make it on I have a list of videos that I should try. I'm trying to make this new series called where I look at a topic that interests me, or if you have any suggestions, that'd be great too. That interests me, and trying to make a video like that, like Vsauce or something like that, because that's pretty cool. I'm always interested in doing stuff like that. And actually, tomorrow we're going to be playing a video game, a couple of friends and I, and we're going to be recording that. And so you can probably see that in a week or two, because it's going to take some time to edit. But yeah, that's about it. If you have any questions, again, let me know in the comments below. If you have any suggestions, again, let me know by subscribing and letting me. It's nice how I sneaked in subscribe in there. Yeah, actually do subscribe, please. That'd really help me out. That'd be awesome. Again, I have all of my Facebook and Twitter and Google Plus and stuff like that, all down there. And maybe if you guys want to follow me, if you think I'm cool enough, yeah, you can do that. If you get to know me, I'm a pretty cool guy. At least I think. Anyways, see you guys in the next one, I guess. See ya.